Good afternoon, everyone. We're excited to have you join us here on the webinar. I'm Katie Raleigh, the librarian at the NOAA Central Library, who will be hosting this. Today we have a NOAA Innovators Series for you, and the person moderating this will be Tiffany House, and I'm going to let Tiffany introduce our speaker today.
Thanks, Katie. Good afternoon. Thanks for joining us today, everyone on the line. My name is Tiffany House, and I am the Commercialization Specialist for the Small Business Innovation Research Program for NOAA. And NOAA provides competition-based awards to small businesses for research and development to translate their innovative ideas into commercial products. The research must meet the objectives of our agency. Today, we will hear from Dr. John Wood of Pisces Molecular. He is in phase 3 of the SBIR program. In this period, his phase 2 innovation moves from lab to market. Dr. Wood will tell us about his NOAA SBIR funded project and how it has been developed. I have the pleasure of introducing Dr. Wood. He is the principal investigator of this NOAA SBIR effort. He founded Pisces Molecular years ago and has carried out molecular genetics research in many areas. Ladies and gentlemen, I give you Dr. Wood.
Good afternoon, everybody. At a brown bag seminar this past summer, Sam Chu from New York gave a nice introduction to the power of environmental DNA or eDNA assays and their increasing use. One aspect that he did not dwell on in his talk was the positive controls for such assays. That's what I'll focus on today, and in particular, the use of plasmid DNA and the advantages that plasmids have over more commonly used genomic DNA controls. Most eDNA assays are qPCR and use TaqMan dual-labeled fluorescent probes, as shown in the diagram on the right. When the newly synthesized DNA strand runs into the probe sitting in front of it, it cleaves the probe, freeing the fluorophore. The fluorophore drifts away from the gray quencher molecule, and we get fluorescence. Most of my talk today will focus on TaqMan assays. However, it is also relevant to cyber-green assays that are used for quantifying high levels of target DNA, such as the harmful algal bloom dinoflagellate assays that I'll talk about towards the end of the talk. Also, we should note that standard curves are used for quantitation in all of these kind of assays. So, how are standard curves made? Take genomic DNA, and you make a series of 10-fold serial dilutions of this DNA. If you then run those reactions in a qPCR reaction and measure the fluorescence as a function of cycle number, as shown here, and this is actually a log scale, so it is the change in fluorescence in a log scale for each of the different dilution standards. Then I can set a threshold, a baseline threshold fluorescence value during the exponential phase of the amplification. And from this threshold, I can then measure or determine the CQ values for each of the standards, where CQ is the cycle at which the fluorescence for a given standard or unknown crosses the threshold. When these CQ values are plotted against copy number on a log scale, as shown here, so X is log copy number, Y scale is the CQ values, I get a standard curve. Because this is a log scale, it is a log linear standard curve. Then I can get similar CQ values for unknown samples and plot them on this same standard curve by changing them down, moving them down to the x-axis. I can actually determine the number of copies in the unknown samples, as shown here. So a CQ of 29.4 equaled 5.54 times 10 to the 1 copies in the reaction. Most qPCR controls have been made with genomic DNA, and the advantages of this are, first of all, it's simple. You take the target organism, you isolate or extract DNA, make the serial dilutions, and you're ready to go. Most of the time, or sometimes, it's actually easy to obtain. For example, one of the assays that we frequently run is to detect rainbow trout eDNA, and it's quite easy to obtain plenty of genomic DNA from fish samples. However, the disadvantages are that if an organism is for organisms that are difficult or toxic to grow, like some of the organisms that cause algal blooms, cyanobacteria or dinoflagellates, then it's hard to get enough genomic DNA, or the quantities are quite limited. It's also difficult to know the exact copy number. Because this is genomic DNA. An example of this is chytrid fungus testing in amphibians. Originally, the qPCR controls for chytrid fungus testing were made from zoospore DNA preps, and they were actually calibrated as zoospore equivalents. The target and the standards were both the Batrachochytrium rDNA locus. However, it was found later that the number of rDNA loci per zoospore varied between 7 and 140 copies, depending on the strain. Therefore, qPCR results using standards from different strains were not comparable at all. Also, the standard curve units, when we're doing serial dilutions of genomic DNA, they're relative. They're 1 times 10 to the minus 1, 10 to the minus 2, 10 to the minus 3, 10 to the minus 4, etc. They are relative units, not absolute. This makes it hard to compare preps between, or compare results, sorry, between different standard preps and between different labs. Another disadvantage is that it's impossible to determine sample contamination. When we're using genomic DNA standards and dilutions that can have between 1 million or even 100 million copies of the target sequence per microliter, even tiny amounts of contamination of those standards into one's unknown samples can give false positives. A frequent response to unexpected positive results or worries about contamination is to sequence it. However, DNA sequencing cannot distinguish between a true positive in a sample and contamination of a sample with a genomic positive controlled DNA, because both are genomic DNA. They have the identical sequence. Because of this, because of all of these disadvantages, we've switched to using plasmid DNA, and I'd like to talk to you about that. First of all, for those of you who are not molecular biology aficionados, plasmids are small circular bits of DNA, in this case about 2,600 base pairs in length, that have a number of common elements. The first is an origin of replication in E. coli. So this allows the plasmid to replicate in bacterial cells, E. coli cells, as the E. coli culture grows. Another item is a selectable marker. In this case, this plasmid has a gene coding for a protein with a function of resistance to the antibiotic kanamycin, so CAN-R resistance. This allows a selectable marker, ensures that as we grow the E. coli in an kanamycin containing media, that all of the cells will actually contain the plasmid. Finally, the last element is what we call a multi-cloning site. It's a series of restriction enzyme recognition sites here that we can use to clone, insert pieces of DNA into the plasmid for any and assorted reasons. So back to plasmid DNA as controls. The disadvantage is that it is indeed slightly more work than genomic DNA. You actually have to construct the plasmid, transform it into E. coli, do a plasmid prep from that, and then do your serial dilution. So it's slightly more work than genomic DNA. The advantages, though, outweigh this. First of all, it's nearly unlimited in quantity. For example, a gigaplasmid prep from a 1 liter E. coli culture can yield up to 15 milligrams of plasmid DNA. For a plasmid that's between 2 and 4,000 bases in length, that equals to more than 10 to the 15th molecules of plasmid. Assuming our most concentrated standard curve dilution is 1 million copies per microliter, and we use 2 microliters in a qPCR reaction, that 1 gigaplasmid prep is enough for 500 million reactions, basically an inexhaustible supply. That means this same plasmid prep, a single plasmid prep, can be used across long periods of time in a given lab and across multiple labs. So 1 plasmid prep to rule them all. Also, for plasmids, the exact copy number is easily determined. The standard curve units are absolute. As shown here in the right, this is a plasmid control that we made for Alexandrium, one of the harmful algal bloom dinoflagellates. We have 7 standards. They range from 1 molecule per microliter, 1¼ molecules, to 1¼ million molecules per microliter. And these are now absolute units. So anyone using this standard can compare their results with anyone else. These are absolute units. It's also straightforward to distinguish sample contamination. I'll delve into this a little more in a minute or two, but basically we can put in a sequence, a target sequence that doesn't match the genomic sequence exactly, so it can be distinguished. Isolating plasmid DNA, the old way, and by old I mean 10 years ago, was to isolate a target sequence DNA fragment, clone the fragment into the plasmid backbone, isolate and grow up the E. coli clone, and finally isolate the plasmid DNA from the culture. This shows a Batrachochytrium dendropatitis, a chytrid fungus control plasmid that we made this way. This actually is a cloned insert of most of the rDNA gene locus into a plasmid. I'll come back to this one later. Nowadays, the new way, given the decrease in cost and time required for producing synthetic DNA, is to basically order the plasmid. We clone by phone, meaning, as shown here, this is a screenshot from GeneWiz, a gene synthesis company that we frequently use. And as shown here in the lower left and expanded, you basically say, I want DNA, you give it a name, and you enter your sequence here, and you order it, and that's it. When you order from GeneWiz, 4 micrograms of plasmid DNA equals between 1 and 10 times 10 to the 11th molecules, depending on the length of the plasmid. And it costs $200 to $800, and it takes about a week to get it. Going back to these numbers, so 1 times 10 to the 11th molecules equals 500,000 qPCR reactions of 2 microliters at a 1 times 10 to the 6th copies per microliter dilution. Again, an almost inexhaustible supply of plasmid DNA from 1 plasmid prep. I don't even have to grow it in E. coli. GeneWiz has done that for me, and they've given me enough that I can use it nearly forever. Because I'm ordering it as a synthetic sequence, I can also put in non-native sequences, as I alluded to earlier. The top sequence is the native sequence, shown here. The synthetic sequence I can vary, first of all, by putting in a completely random sequence. The base is shown in red here. Or I can insert a restriction site where it didn't previously exist by changing a couple bases here. I now make a recognition site for the enzyme EcoR1, or for a pre-existing restriction site here, CLA1 in the native sequence, I can alter bases and remove that restriction site, so it's no longer present in the synthetic sequence. This allows me to distinguish the synthetic sequence in my plasmid from native genomic sequence, either by sequencing it or by cutting it with the restriction enzymes and showing that the restriction enzyme pattern is now different. Another advantage of this synthetic sequence is I can put multiple targets in one plasmid. Shown here is an insert in a control plasmid for detecting for use in assays detecting 4 different species of salmonids: grayling, chum salmon, Chinook salmon, and coho salmon, all in one plasmid control. So how do we quantify these qPCR control standards? The typical way has been to make a genomic DNA prep, measure the concentration of the genomic DNA in a spectrophotometer or a fluorimeter, and then make the serial 10-fold dilutions that I talked about earlier, and calculate the standard concentrations as 10-fold decreases from the concentrated stock. The problem with this is multiplicative errors. In each of the dilutions, there's an error, and of course, by the end, where I'm at the most dilute, these errors have multiplied. The greatest error is in this lowest concentration standard, which is the standard that I want to use, or that we use to determine the sensitivity and limit of detection of the assay. So we have decided this is not really accurate enough. As an alternative, we've done a similar procedure, making plasmid DNA preps, getting approximate concentrations, only approximate, and then running what we call a Poisson P0 experiment, where we have made 88 replicate qPCR reactions with the lowest standard dilution, somewhere between 1 and 5 molecules per reaction. At this low number of molecules in a reaction, in the template that we add to a reaction, we expect stochastic sampling error to cause some number of these reactions to fail. By doing a large number of these reactions and collecting the results, you can see that many of the reactions gave CQ values of about 34 to, I think there's a 46 here, a really late one, but many of the reactions failed. We count up the number of failed reactions versus positive reactions, and in this case, 47 of the 88 reactions failed, or about 53%. 53% then represents the P0 fraction, x equals 0 fraction of a Poisson distribution. So P0 equals 0.534. And for the fractions in the Poisson where x is greater than 0 are just shy of 47%. From this, and assuming a Poisson distribution of the molecules, we can calculate that the mean value of the molecules for this dilution, or in these reactions, is 2.5 molecules per reaction. Because we use 2 microliters of template per reaction, then in the dilution itself, the actual concentration on average is 1.25 molecules per microliter in the dilution. And this now then has quantified the most dilute standard, the one that is most important for determining the sensitivity and limit of detection of the assay. And this is the one most critical for that. Next, we looked at stability. For any controls, DNA or otherwise, of course, stability is a big deal. You don't want them changing over time. So we ran a series of experiments to look at our Alexandrium plasmid dilutions. Again, these are the 7 dilutions ranging from 1.25 molecules per microliter up to 1.25 million molecules per microliter. They are shown along here, along the Y scale, each of the 7 standards. And being good molecular biologists, we kept these at -20 degrees C in a freezer and in the dark. And every week we pulled them out, thawed them, and ran a qPCR over the course of 5 weeks. And what you can see is no change in the CQ values shown on the Z-axis for any of these standards across 5 weeks, except one dropout in dilution number 7, the lowest numbers of molecules per microliter, probably sampling error right there. If indeed the plasmids had been degrading, if the DNA had been degrading, you would have seen an increase in the CQ values. Fewer molecules means later amplification, greater CQ value. We saw no change at all. But this is, again, kind of the standard for molecular biology, stored in the freezer. Next, we actually tried other storage conditions. First of all, down in this lower left graph, we did the same thing, stored at -20 degrees C, but we thawed it daily. So a freeze-thaw cycle every day. All good molecular biologists have been taught that freeze-thaw cycles can be deleterious to DNA molecules. So again, we ran a qPCR reaction every week. However, between those weekly qPCR runs, we did a freeze-thaw cycle every day on all 7 standards. This is a different set of standards, of course, than that. Again, little or no change in the CQ values over the course of 5 weeks, except again in the most dilute standard, number 7, we had 3 dropouts where they failed to amplify. We think these are indeed the stochastic sampling error. This is good molecular biology practice. This is where you're really busy and you're running the standards every day. In the upper right here, we actually kept the samples, another set of standards at 23 degrees C or room temperature. And this would be if you took your controls out, ran a qPCR, and forgot to put them back in the freezer. You left them on the lab bench. So these were again at 23 degrees C over 5 weeks. Again, yet again, no changes in CQ values at all. And finally, we did an even more brutal treatment. We actually stored a set of standards at 37 degrees. The lab example of this would be that you were ready to run a qPCR reaction and you took your standards out and they were frozen, so you stuck them on a heat block to thaw them more quickly, and then you forgot and left them on the heat block at 37 degrees. Even at 37 degrees, over the course of 5 weeks, there were no changes in the CQ values for any of the standards. Therefore, these plasmid DNA control standards are exceptionally stable. Next question that we wanted to ask was are there any strange or deleterious effects of putting multiple qPCR targets on one plasmid? Are there any effects on amplification? Shown here, diagrammatically, is a target A amplicon and a target B amplicon all on the same insert. And one of the worries was that forward primer A, rather than stopping at the end of amplicon A, could keep going, the strand could keep being synthesized all the way into target B, and reverse primer B into target A to give us what we call a run-on amplicon. Obviously, run-on amplicons would not be good if we're trying to quantify the amount of target A or of target B independently, because this one actually contributes to both. It's using forward primer A, reverse primer B. It would distort the quantification of amplification of either of the individual targets. So we designed a plasmid control to test this. Here are 2 separate target amplicons. The first one from primer 793 to primer 794. The probe for this one is a FAM probe. And the second amplicon is from primer 795 to 796. It has a Texas red probe. 2 other things to point out here are there are AFOL3 restriction sites at either end of the synthetic fragment and a SCA1 restriction site between the 2 amplicons. So when we cut the DNA out of the plasmid with AFOL3, we get both amplicons on one molecule. When we take that fragment and cut it with SCA1, we separate the 2 amplicons from each other. So shown here, this is a single fragment. The AFL3 digest is a single fragment for both amplicons. The SCA1 digest is multiple fragments for the two different amplicons. And these are the CQ values when we amplify them under different conditions. We run them in qPCR reactions under different conditions. First of all, if we run the upstream amplicon only PCR, so only primers 793 and 794, or the downstream only primers 795 and 796 with the Texas Red probe, we get particular CQ values. Or we combine it all and we run it in a multiplex reaction, both sets of primers and both sets of probes. As shown here, the CQ value for the AFL3 digest, so both fragments on, both amplicons on the same fragment, we get a CQ of 19 3/4. We get essentially the same CQ value when we run in extra primers. So the primers 795 and 96 in the reaction are not affecting or altering the amplification of the upstream amplicon. And if you compare all 6 pairs, so the upstream amplicon by itself or with the downstream primers, no change at all, or the opposite, again, the downstream amplicon by itself or with the upstream primers, no changes in CQ value at all. I haven't talked about this before. G-blocks is actually a separate set of where we had these two fragments, these two amplicons synthesized completely independently as completely separate fragments. I'll talk about this more about this in just a moment. So back to why it's not advancing. Okay. And so no differences in CQ values for targets together on one molecule in multiplex or on separate molecules or in singleplex. Now back to G-blocks. I talked about this before. Shown on the left here is a plasmid with a very complicated insert targeting a whole series of assays and attached to the plasmid backbone. There's no particular reason that one needs to have this synthetic fragment integrated into a plasmid backbone. You can have just the fragment synthesized by itself. We frequently buy these from Integrated DNA Technologies, IDT. IDT calls these G-blocks. So we could just as easily order the synthetic fragment all by itself. Several issues with this are, first of all, it's in limited quantity. What you get is what you have. Because this can't be replicated in E. coli, whatever you get from IDT is what you use. When you run out of it, you have to put in another order from IDT. Plasmids, however, if I use up the 4 micrograms that I got from GeneWiz, I can transform the remaining amount into E. coli, grow up a large E. coli culture, and like I said, with a plasmid prep, make inexhaustible quantities of this. Another unexpected problem with this that we hadn't really anticipated was synthesis issues. It's hard to see at this scale, but there are actually 4 different Ranavirus assays on here. All of these assays target the major capsid protein of Ranavirus and they're in different regions of the major capsid protein, and therefore they are related qPCR assays, if you will, and they have homologous sequences from one to the next and onward. When we asked IDT to synthesize this fragment, they refused. They turned us down. They said there are too many homologous sequences here. It interfered with their synthesis process. I'm not quite sure why IDT can't make it, but GeneWiz didn't have any problem. But that is a problem with, at least that can occur with G blocks. More important is that anecdotal evidence suggests that G blocks are less stable. We think this is due to low-level exonuclease activity chewing off the ends of the molecules. And you can see in a G block, if we chew back the end of this fragment here, we're very quickly going to destroy the primer binding site for this particular assay, or chewing at this end will destroy it for this assay, meaning then the controls won't work anymore. For plasmids, on the other hand, we actually linearize them at these DRA1 sites shown here. So any exonuclease activity would have to degrade hundreds or even thousands of bases before it got into the synthetic fragment and started interfering with their use of a control. So G blocks appear to be less stable. We're actually currently running experiments to test this for real. I've shown you this plasmid before earlier. This is the BD control plasmid with the rDNA or rRNA gene locus in it. This one, like I mentioned, has targets for multiple BD assays. This allows easy comparison of different assays used in different labs. However, it turns out a few years ago that there's a second chytrid fungus to worry about. This one, BD, infects many or all amphibians. There's a new one, Batrachochytrium salamandrivorians, which, as you might imagine, primarily affects salamander species. Again, apparently responsible or potentially responsible for the die-off of salamanders, at least in Europe and Southeast Asia. It has not made it to North America yet, but everybody's worried. So since this is one to worry about, we've now made a plasmid that control plasmid that works for both. Here's the BS amplicon assay designed by a guy in Holland. Here's the BD Boyle amplicon that I showed you in the previous plasmid. Here's the Pisces BD amplicon as well. So now this control can be used for multiple species of chytrid, only one control, and or a multiplex assay. Also, because these targets are all on the same fragment, they're guaranteed equimolar. If we'd actually made these as two different genomic preps or even two different G blocks, then we would have had to go to a lot more work to guarantee that they were equimolar in concentration. Putting on the same fragment in a plasmid, that's taken care of. But amphibian world is still has even more problems. Ranavirus is another pathogen implicated in many of the amphibian die-offs that have occurred worldwide. So let's make a control plasmid that takes care of that as well. And I'd shown you this picture smaller previously. Here it is blown up. You can see here's the BD assays. Here are the B.sal targets, and then 1, 2, 3, and 4 Ranavirus assays. Targets for all of those. Ranavirus is an intracellular virus, therefore the number of Ranavirus copies or DNA molecules that you get in a prep or from a sample, of course, is dependent on how many cells you actually got in the prep as well, and that will vary between, depending on whether it's a tissue sample, a blood sample, a toe clip, or a skin scrape or skin swab. So we added in target for yet another assay called an ultra-conserved non-coding element, EBF3 Napoleon. It has been shown to be a single copy sequence in all amphibians, and one copy per haploid genome. Therefore, I can use this to normalize the results for my Ranavirus assays to the number of cells in my DNA prep from a given sample. And yet one more element we put on here is a detection fragment. This is a synthetic fragment we designed at Pisces some years ago. It does have primer and probe sites that are unique for this fragment and completely different from any known genomic sequence. So we can use this assay to determine the presence of plasmid DNA. So the EBF3 element is the internal control for cell number. The plasmid detection fragment allows us to determine whether we have plasmid DNA. This is even simpler than sequencing a restriction digest. It does allow us to determine sample contamination with plasmid control. And as part of our work with NOAA on the SBIR grants, we've been developing controls for Alexandrium species. These were species that were predominantly identified by Wayne Littaker at the NOAA South Carolina labs in Charleston. There are 7 different identified species. This control will work for all of those assays, and again, it has the detection fragment at the end. Not stopping with Alexandrium, we went on to Gambierdiscus, another harmful algal bloom dinoflagellate. There are a whole series of different identified species of Gambierdiscus. In fact, this plasmid now targets 11 different species, or can serve as a control for 11 different species-specific assays, plus 2 genus-specific assays. There's basically no limit to how much you can put on a plasmid control. Again, finally, the detection fragment as well. Plasmid controls can also work beyond just simple qPCR assays. Shown here is a 16S plus bacterial 16S plus fungal ITS control plasmid for next generation sequencing. In many metabarcoding studies, especially with environmental samples, it's crucial to be able to compare the extraction, amplification, barcoding, and read efficiencies between samples. This is generally done by adding known small numbers of control 16S or fungal ITS targets at the beginning of the procedure and measuring the overall procedural efficiency by how many reads of each of these control sequences you get at the end. The control sequences can't be native sequences because then they couldn't be distinguished from true reads from sequences actually in the sample. So in the same way that we did for the plasmid controls I've already shown you, we designed synthetic bacterial or ITS sequences which have different non-native sequences called here synthetic, here randomized, so they can be distinguished from native sequences. Because we want these synthetic sequences to amplify no better and no worse than native sequences, we were careful to keep the length and the average GC content the same for these synthetic or randomized inserts as compared with native sequences. And we also edited these to eliminate any repeat or palindromic sequences which could form unwanted secondary structures during amplification and therefore change the efficiencies. We also included yet again the detection fragment because we want to accurately quantify the number of molecules in a control plasmid prep. This, of course, is crucial because we want only enough reads from the control plasmid to compare between samples. Any more than that just wastes the finite number of reads one gets from an NGS flow cell run. And one less plasmid that we're actually currently designing and constructing, we call it a DNA Degradation Detection plasmid. One of the still not very well understood aspects of environmental DNA sampling is how long eDNA persists or not in the environment, or the best way to preserve it after a sample is collected. Should the filters be frozen, desiccated, in ethanol. So we designed this plasmid to help address these questions. As shown in the diagram, we designed a sequence with one forward primer and multiple reverse primers and probes at increasing distances from the forward primer. We've also added a unique restriction site immediately distal or outside the most distal primer site here. So when this plasmid is cut and linearized at this restriction site, any exonuclease activity will degrade or remove the reverse primer binding sites in sequential order. On this end, there's no worry because we have the entire plasmid backbone, so exonuclease activity is never going to get into this forward primer site. So with this plasmid, we'll be able to test the effect of different sample preservation or extraction techniques. And see which, if any, of those preservation or extraction techniques have any effects on the longest sequence or all the way to shorter sequences as the exonuclease activity or any other DNA damage proceeds closer to the forward primer. And referring back to my illustration earlier of how much plasmid DNA we can make in a single plasmid prep, we might even be able to use this DDD plasmid to test the stability of the DNA in the environment. Because the gigaplasmid prep yields 15 milligrams of plasmid DNA or 10 to the 15th molecules, we could potentially even add it to a lake or a stream and have enough molecules to recover in a filtered water eDNA sample and directly address the idea of how long DNA molecules persist out in the environment. So with all of that, I'd like to go over conclusions. Plasmid DNAs have a number of advantages as qPCR positive controls. They are easy and inexpensive to produce in basically unlimited quantities. They're exceptionally stable. They can be quantified absolutely. They can be made easily distinguished from the native genomic sequences, and they can be made with targets for multiple assays or multiplex reactions. And with that, I would like to acknowledge this. Indeed, much of this work was supported by NOAA SBIR grants as shown here, and I'll be happy to take any questions. Thank you.
Thank you so much, Dr. Wood. That was very informative and very much over my head, but I hope our online audience has some questions for you. If you do have a question, please place it into the question panel. If we don't get to it today, we will email that to Dr. Wood and he will respond to you offline. I am not seeing any questions right now. Let's give everyone a minute to formulate their thoughts. Okay, if anyone does have a question, you can see Dr. Wood's email in the corner of this slide here. And if you would like to revisit this presentation at a separate time, the library will be uploading this to our YouTube channel because we have recorded it. And thank you all for your afternoon, your time. Thank you, Dr. Wood.
Thank you.
Oh, are you, okay, we do have a question. Someone responded. Stop us real quick. The question is, do you think the delayed CQ of the G blocks was caused by the exonuclease reaction?
Actually, no, I forgot to mention that. I think that was the quantitation of the G block, the amount of DNA that we got from IDT was not as accurate. We were actually trying to dilute it and put in the same number of molecules as our plasmid because we did not do a Poisson P0. I don't think the quantitation was as accurate, and that's why the later CQ activity.
Okay, thank you. We have a few more questions. Besides G-blocks, did you ever investigate making your own standards via PCR? As in, couldn't you amplify a longer region from your gDNA, quantify it to get copy number, and run it as a control instead?
Sure, that's a possibility. However, even a PCR reaction won't give me anywhere near the same number of molecules as from a plasmid prep. I don't know that you can really do a preparative PCR. I mean, theoretically, yes, it's possible. It's easier, faster, quicker to just order it from GeneWiz or companies like that.
Great, and we have one more question. Does the plasmid have to be linearized before use? And if so, doesn't that affect stability?
Again, a point that I skipped through too quickly. Yes, all of our plasmids are linearized. There's been studies both we've done and other people have done that plasmids are typically supercoiled as they're isolated from E. coli. Supercoils do adversely affect PCR amplification efficiency. So we do linearize the plasmid, and we linearize it at those DRA1 sites. I can see if I can go back to that slide. Find it here. Show it there. Yeah. From current slide. Okay, it's not getting on my screen. But yes, we do linearize the plasmid with DRA1 at sites that are on the opposite side of the plasmid backbone from the insert. So any exonuclease activity there would actually have to traverse thousands of bases, at least 1,000 bases, before it would impact or start degrading the insert sequence. Therefore, no exonuclease activity that we can detect. Again, the stability experiments, those were all linearized plasmids. Even 5 weeks at 37 degrees, we had no problem. I didn't show you, actually the experiment didn't end at 5 weeks. We actually left the samples for 13 weeks total, and there was no degradation, no increased CQ values for any of the plasmid dilutions across 13 weeks at 37 degrees.
Okay, thank you. We do have one last question. What about the accuracy of the plasmid sequences? Can there be mistakes?
Yes, there can. However, GeneWiz and all of these reputable places, they sequence it. So they send me the sequence of the DNA of the clone that they made for me. So the sequences are exactly what I ordered.
Okay. I think that is the end of our questions. Thank you so much, Dr. Wood, and thank you everyone online for asking your questions and sticking around. I hope you all have a wonderful afternoon.
Thank you all.